Good afternoon, everybody. Thanks for attending today's X-Talks webinar. Today, we're going to discuss the EU's new medical device regulations that go into effect in May of this year. We'll also describe how a third-party analytical lab, such as Eurofins EAG, is able to help support your company's efforts in complying with the new regulations as they evolve into the 2020s. We'll commence by providing a brief outline of today's presentation. First, we'll discuss the evolution of the EU's medical device regulations, how specific events emphasize the need for stricter and more comprehensive directives governing patient safety across Europe. We'll then highlight some key changes from previous regulations that represent areas where companies may need to fill in the gaps in their medical device information portfolios, both from a technical documentation standpoint, all the way through a more comprehensive testing program to support a device's biocompatibility through toxicological evaluation. We'll spotlight some expectations for medical device manufacturers in order to demonstrate compliance. Next, we'll cover reasons why companies would choose to use an analytical lab. You may find yourself in one of these categories, or it may help broaden your considerations of how they might be helpful to companies like yours in the future. Next, we'll provide a brief introduction to who Eurofins EAG Material Science is and how our mission and identity is driven by different key industry sectors, each with their own unique set of essential requirements and key challenges. We'll also list particular service offerings that speak to the needs of the pharmaceutical and medical device community. Finally, we'll provide some case studies based on analytical investigations of a medical device which address some of the requirements for ISO 10993, Parts 18 and Part 12. So in 2017, the EU's revamped medical device regulation was published, which expanded the scope of regulations and requirements that currently govern medical device manufacturers under the Medical Devices Directive. The timeline provided by the MDR was 3 years, which extends until the 25th of May of 2020. The driver behind these new regulations was a desire to increase the overall health, safety, well-being of patients, in addition to incorporating technological advances in scientific capabilities for testing. So there actually was a notorious health scare that originated by a company in France who made breast implants beginning in the early 1990s that had lasting global repercussions. This is considered one of the drivers for change and reform among the European medical device community that prompted the need, drafting, and implementation of the new MDR. The company, called Polyimplant Prosthèses, embarked upon making different types of breast implants around the same time that the US FDA halted sales in the United States. PIP produced hydrogel, saline-filled, and originally medical-grade silicone implants. Audits of the facility in 2000 found non-conformances with GMP practices, and hydrogel implants were removed from the market due to the company's inability to prove their safety. Shortly thereafter, as a cost-cutting measure, PIP began using industrial-grade silicone, not approved for medical-grade use, in their silicone breast implants. Legal problems later ensued for the company, ultimately with doctors reporting device failures due to rupture at twice the typical industry average rate. The company went bankrupt and the French Medical Safety Agency recalled the implants. Upon review, it was discovered that the implants contained only 10% of the approved medical-grade silicone and 90% of the unapproved technical grade. In 2011, the French government made the recommendation that up to 30,000 women consider removal of these tainted implants. The end result of this 20-year ordeal highlighted the need for an extensive overhaul of the regulations that govern the medical device industry. As a result, the European Commission proposed and drafted a new regulatory framework called the European Medical Device Regulation, which went into effect in 2017. The following points were emphasized in the new regulations as a result of that incident. Inclusion of cosmetic aesthetic devices under the broad term medical device. creation of a national registry that requires manufacturers to register themselves and devices in a central database to assist in traceability, imposing restrictions and bans on financial incentives and aggressive marketing in favor of patient safety, and establishment of a centralized database to document adverse health events associated with devices and their associated corrective actions. As a result of the implementation of the new MDR, if a company desires to market their device in Europe, they'll need to understand some key changes from previous directives. Some principal differences are elimination of the grandfathering mechanism. This necessitates that all medical devices currently on the market must apply to be accredited, regardless of whether or not they were previously approved under earlier directives. Empirical evaluation and testing that demonstrates that devices are biocompatible in alignment with ISO 10993, and not just reliance on previous historical safety data. Documentation showing evaluation of medical devices for the presence of carcinogens, mutagens, and reproductive toxic substances. The new MDR requires a more comprehensive data demonstrating biocompatibility to ensure both the medical device itself and the individual components and raw materials are safe. The expectation that the MDR sets forth for medical device manufacturers is the following if they intend to sell devices in Europe. The need to ensure proper documentation is readily accessible. The need to possess a comprehensive portfolio for every material that's used in the manufacturing of devices. They need background documentation on the composition of the device, a list of materials, specific grades, and how the device is assembled. They need records of replacement or substitution of materials over the product lifetime. And finally, they need documentation on biocompatibility and data that supports this conclusion. Additional expectations to be in compliance are the following. The need for a gap assessment in order to bridge the original testing performed and compare it with ISO 10993. And then after the comparison, to come up with a list of additional testing that may need to be performed in order to bring the level up to the ISO 10993 standards. Since biocompatibility testing may be costly and time-consuming, companies may need to strategize how they may effectively group devices by chemical families based on materials that they are composed of and the applications of devices in order to streamline the testing. Arranging for an initial risk assessment that provides specific information about intended use for the device. For example, is the contact with the patient limited, prolonged, or long-term? Based on this, it could require either a simulated use chemical extraction or exhaustive extraction in order to be able to calculate the AET or analytical evaluation threshold in order to help understand exposure to patients. Toxicologists can issue a toxicological risk assessment based on the chemistry of the material, the concentration, and the exposure duration, as well as the type of patient contact, such as oral, dermal, or implanted in the body. Focusing our lens on our medical device and pharmaceutical clients, in addition to performing testing to satisfy the EU's new MDR, there are other types of testing that we perform to address some of the key industry challenges that medical device and pharmaceutical clients face. Testing may need to be performed to ensure product quality. Does my product meet its provided specifications, and are my suppliers providing me with materials that actually are what they say they are? Safety. Is my product safe? If contamination is found, what is it? What level or concentration is it? And is it of toxicological concern? Regulatory. Does my product meet the standards specified by specific agencies? And outsourcing. Our company's quality system adheres to a strict set of standards. However, for testing that requires outsourcing to a third party, how do I guarantee that my outsource partner is providing high integrity data that meets or exceeds our company's current standards? So Question for you. How many of you have actually outsourced testing to a third-party lab? If you have or if you have not, here are some of the reasons that you might consider doing so in the future. Maybe you don't have an R&D lab. It could be due to the size of the company that this need would need to be outsourced, or it could be due to financial consolidations that an in-house R&D lab could have been phased out. Some of our clients have informed us that they do have an analytical lab, but that the lab is so backed up that they choose to outsource in order to be able to have access to timely or expedited results. Some clients choose to work with us because it provides them with a diverse range of analytical capabilities that they don't have in-house. Others admit that they actually have the actual techniques in-house, but that they prefer the expert data interpretation from a team of scientists and engineers that specialize in problem solving. Finally, there are definitely some situations where companies actually require third-party or independent laboratory testing to ensure integrity of the results with no internal bias that could be influencing the results to turn out a certain way. As the global leader in materials and engineering sciences, EAG supports thousands of clients with facilities located in the US, Europe, and Asia. Eurofins EAG Material Science offers multidisciplinary approach to problem solving, bringing together state-of-the-art instrumentation and industry experts in chemistry, material science, metallurgy, plastics and polymers, microscopy, semiconductors, reverse engineering, and medical device testing. In addition to in-house techniques, EAG has relationships with partner labs to provide additional services such as toxicological risk assessment, microbiological testing, and other specialized testing per client request. An advantage of being a worldwide solutions provider is we have access to a big toolbox. So these are just some cartoons that kind of illustrate the alphabet soup of analytical techniques that we have access to. As a result, we have the tools and the know-how to design a customized study in order to help you answer your question. So you may be asking yourself what types of services may be relevant to you in your industry. Here are some examples of common studies requested from pharmaceutical and medical device clients. Particle size analysis, to determine both the number and size distribution of particulate contaminants within a particular device. Particle material characterization, in other words, understanding the chemistry of the particle. Having information about the overall number and size distribution of particles may not be enough to guarantee patient safety. Having access to information on what the particulates are composed of from a chemistry materials perspective will allow a toxicologist to assess patient risks if they are exposed to a specific level of types of materials given specific clinical duration and route of exposure. Polymer characterization and degradation studies. To help confirm the chemical composition of a polymeric material used in a device and determine its chemical compatibility or robustness under expected clinical conditions and degree of degradation over time. EU regulations and ISO 10993 Part 18 reverse engineering deformulation of material to identify its chemical constituents and quantify their concentrations in the formulation. We've developed cleaning validations to assess effectiveness of a sterilization or cleaning process. We've also performed raw material characterization, such as performing purity assays for known raw materials, screen for heavy metals, phthalates, etc., as part of REACH, RoHS, or other compliances. And finally, we support biocompatibility studies requiring material characterization. So now I'd like to introduce our case studies. So for our case study today, we're going to be discussing testing on a medical device that comes into skin contact with a patient for hours of duration at a time. So just to give you some context for this, a medical device company came to us to characterize one of the products that has been on the market. The product's description in terms of its chemical family was vague. It's possible that it could be a simple formulation, but it is also possible that full disclosure was not being provided. The company came to us to identify what components were present and quantify them down to the level of 0.1%. So the sample that we're going to be discussing for the case study is actually made up of two parts. The first is a hard, clear polymer plastic that bridges the gap between the patient and other equipment associated with the device. The second part of the medical device is a soft, opaque polymer plastic that makes contact with the patient. And with that, I'm going to turn this over to my colleague, Dr. Scott Graham.
Thank you for the introduction, Carolyn. So as Carolyn just mentioned, we have a lot of customers who may know something about their medical device or the materials that are involved, but it may not be enough to satisfy the MDR requirements. So this is where EAG can help you try to determine, identify, and quantify the different components in various sorts of medical devices. So we'll be starting off by looking at the hard part of the polymer in this medical device. So for each of these case studies, I'll try to give a quick overview of the analytical tools that we will use to characterize these materials. And like most things in the world, there's kind of some pros and cons with every technique. So most complex problems can't be solved using a single technique alone. So to start this one off, we'll be characterizing the hard part with FTIR spectroscopy. This is a way to give us a bulk chemical composition or identify the types of functional groups that are present in the sample. Here on the top, we have three spectra. The top one is of the actual isolated hard part of this medical device. And then we have some spectral library matches for a polycarbonate PET copolymer. And comparing the spectra, we can see that there's a lot of spectral overlap and similarities between them. However, if we zone in and look at this peak that's at about 1740 in our sample, it doesn't quite match up to the peak in our reference spectra that's at about 1720. This corresponds to an ester-type functional group that's on the polymer backbone. So this may suggest that the material is some sort of a polycarbonate polyester mixture, but it may not actually be PET. To dig a little bit deeper, we'll use another analytical tool called NMR spectroscopy. NMR stands for nuclear magnetic resonance. And this is actually a technique that some people may be a little bit familiar with and they may not even know it. So this is the same technology that's used in medical imaging. So the word N, nuclear, was actually dropped from the phrase NMRI to just become MRI because the word nuclear has somewhat of a negative public connotation. So this is a spectroscopic method that can allow us to dig deeper into what the chemistry of this polymer is. It's a solution phase technique, which means that we need to be able to dissolve our sample. And for NMR, we need to use a deuterated solvent so it doesn't create additional signals in the sample. Using the information that we gained from FTIR spectroscopy, we're able to look up materials that can be dissolved with an appropriate solvent. And for this, we use deuterated chloroform. We're also able to reference literature to look at a normal spectrum of PET. And when we compare that to our sample, we realize that there is a prominent chemical shift at about 4.4 ppm in PET that was not present in our sample. So again, this kind of further solidifies the minor differences that we saw between the sample and our reference spectra by FTIR spectroscopy. Consulting the literature once again, we were able to successfully identify the polymers that are present in this sample using NMR. This material is consistent with a polybisphenol A isophthalate terephthalate copolycarbonate. We have three peaks that are labeled red that are due to the isophthalate residues, and we have one peak that corresponds to the terephthalate. We can see that there's some differences in the signal intensities, so right now we would believe that the isophthalate seems to be the major species with respect to the terephthalate. On the bottom right of the slide, we see two different structures. On the top is the terephthalate, and on the bottom is the isophthalate. For the terephthalate, there are 4 protons that are labeled F. These are all chemically equivalent due to the symmetry of the molecule, and they only collapse to create one peak in the sample spectrum. On the other hand, the isophthalate, which also contains some degrees of symmetry, but not quite as much, end up producing 3 signals in our spectrum. So we were able to identify these using literature, and we were also able to identify the remaining peaks in the sample spectrum. These are going to be due to the BPA residues in both of these polymers, as well as from the polycarbonate. And these are going to be the protons that are labeled D, E, and G. So what does the structure of this polymer look like? If we take our BPA terephthalate, our BPA isophthalate, and our polycarbonate, we get the structure that you see at the bottom. So this is our polybisphenol A, isophthalate terephthalate, copolycarbonate. When we connect these, we use the nomenclature N, M, and L to denote the relative ratios or the amounts of these different monomers relative to one another. So we can dig a little bit deeper into the data and determine what this ratio is. NMR is a quantitative technique. So for this, we can use the peak area integrations, which give rise to these signals. And if we know how many protons in a specific monomer create the signal, we can determine the relative ratio of these in the polymer. So if we're looking at what is the ratio of the isophthalate to the terephthalate, we can divide the peak integrations by the number of protons giving rise to that signal to determine there's about 13 times more of the isophthalate relative to the terephthalate. We can perform a similar calculation for the polycarbonate, but most people are usually used to thinking of things in terms of weight percents versus how many monomers are present relative to one another. Using the formula weights of these different monomers, we can convert the molar ratios or monomer ratios to weight percentages, which are a little bit easier for most people to understand. When we perform these calculations, we are able to conclude that this sample contains about 3% of the terephthalate, 43% of the isophthalate, and 54% of the polycarbonate. So what do we learn from this first case study? Analyzing the hard part of this medical device was able to let us know what the exact composition was. So we got a good idea what it was from FTIR, that it was some sort of a polycarbonate polyester mixture, and that PET may not be exactly what we're looking at. Using spectra and literature databases, we were able to determine using NMR spectroscopy that it's actually a bisphenol terephthalate isophthalate polycarbonate mixture. So now this customer could take this data and file it in their MDR and be confident that they know everything they need to know about this polymer's composition. They can add this information to their internal dossier, and they can also monitor this composition over time for quality control and consistency. For the second case study, we'll be looking at the soft polymer. So as Carolyn mentioned, this is the part of the medical device which makes contact with the patient. Just as we did in the first case study, we'll start off by analyzing the sample using FTIR spectroscopy so that the spectrum looks a little bit different. And if you are a spectroscopist, you may recognize this. So this will bring us into our second poll question. So the good news is if you didn't have an idea what that was, or you maybe had an idea, or you have no idea what you're looking at, that's what EAG is for. We're here to help customers solve their complex needs using chemistry and other related techniques. So if we look again at our material, this is the soft part of the medical device. And on the top, we have our sample, and on the bottom, we have our spectral library match. So this is coming up as a silicone rubber or polydimethyl siloxane, also known as PDMS. And when we compare these two spectra, unlike the first case study, this appears to be a very, very good match. So we don't see any additional peaks that are present in the sample spectrum that are present in the spectral match or vice versa. This leads us to believe that it's a homopolymer, meaning it only contains one sort of monomer unit. So what other techniques can we use to characterize this material? Next, we analyzed it using thermogravimetric analysis, or TGA. This technique involves heating a small amount of the sample, usually just a couple milligrams, under a nitrogen atmosphere from ambient conditions up to 800 degrees C, or about 1500 degrees Fahrenheit. As the sample heats up under nitrogen, small volatile components can evaporate or volatilize, and this is observed as a mass loss. As you reach higher temperatures, you can start to degrade and decompose the polymer, and again, you can observe this as a mass loss event. So looking at this sample, what we see is it contains about 2% of volatile to semi-volatile materials up until about 350 degrees. This could include small cyclic siloxanes like the D4 that's shown in the spectrum. Next, we see two larger thermal decomposition events of about 15% each. So this is actually the PDMS backbone breaking down into smaller fragments and volatilizing and leaving the sample pan. The most important piece of information that we'll take away from this is that after heating the sample to 800 degrees C, 67% of the initial sample mass still remains on the balance. At this point, we can usually conclude that this is going to be some sort of an inorganic material, as most organic compounds decompose well before 800 degrees. So we suspect that this is some sort of filler in this polymer, but what is it? For this, we'll again introduce another technique known as EDS, or elemental dispersive X-ray spectroscopy. This is a technique that allows us to detect what the elemental composition of the sample is. And just as we suspected, we aren't seeing any carbon in this residue. All the material that we're seeing is silicon and oxygen. Some of the overlap in these elements is also present in PDMS, but PDMS can't survive at these temperatures. So the presence of silicon and oxygen is consistent with silicon dioxide, or silica. And so this is what's used to fill the polymer. What's not shown here is we also ran a pure PDMS standard alongside the sample, and after heating it to 800 degrees, only 0.5% of the sample mass remained. This gives us further confidence that we're indeed detecting a filler and not just oxidizing the siloxane species to silica. The final technique we'll take a look at is pyrolysis GCMS. This involves heating the sample as well, but in two different steps. And instead of observing the mass loss as a function of temperature, we'll actually be identifying the volatile components and gases that come off the sample as it's exposed to these elevated temperatures. The first experiment is a desorption experiment. This heats the sample to 250 degrees C. This will volatilize small organic compounds as well as any additives that may be in the polymer, and we're able to detect them in their original form because most organic species can survive at this temperature. What we detect are a bunch of different siloxane-related species, which again is consistent with the presence of PDMS that we are able to detect by FTIR. The second step is the pyrolysis step. This heats the sample at a higher temperature at 600 degrees C. At this temperature, most organic components tend to break down and decompose into smaller products known as pyrolysis products. Pyrolysis data interpretation can be challenging considering that the molecules you're detecting may not actually be present in the polymer that you're looking at. So looking at reference spectra in literature is a must. Here again, we detect some common small pyrolysis products, but again, the primary peaks that we're detecting are all siloxane related. So this again further solidifies the fact that we don't think there's any prominent additives as well as any other copolymers present in the soft part of this sample. So what can we take away from our second case study? Again, FTIR proved to be a great technique right off the bat to determine the bulk chemical composition of this, and it looked like it was a silicone rubber. From TGA and EDS, we saw that this had a very low volatile content and a high amount of inorganic fillers, which with the EDS analysis led us to believe that it's silica. So it's a silica-filled silicone. Finally, the pyrolysis and GCMS, we didn't detect any prominent additives in our desorption step, and the silicone is really the only species that's present here. So now the client can take this information, cross-reference their other materials and safety data sheets to confirm that the product matches their expectations. For our third and final case study, we'll be looking at an extractable and leachable experiment. Extractables and leachable components are materials that can be extracted from a bulk chemical material such as a polymer. To do this, we expose the sample to solvents to kind of pull them out of the material they started off in into a solvent so then that we can detect them. Extractables can be thought of as anything that can be removed from a solvent through interaction with something that's designed to swell the polymer and actually aid the migration of these small molecules into the solvent. For this, we want to choose a solvent that's going to maximize our extraction efficiency without actually degrading or dissolving the polymer. So this is going to be a very comprehensive extraction. The sister experiment is the leachable experiment. This can also be thought of as pulling things out of the polymer, but this time we're going to be using a solvent that's similar to what it may encounter during its intended function. So usually this is going to be somewhat of an aqueous-based extraction, considering most medical devices are around humans and we're made up with a lot of water, so this makes sense. So this is the real world scenario. When we perform these extractions, we follow the guidelines of ISO 10993 Part 12, Biological Evaluation of Medical Devices, Sample Preparation and Reference Materials. So to perform these experiments, we use 3 different solvents that cover a wide range of polarity. So we have polar solvent, which is water, a semi-polar solvent, which is isopropyl alcohol, or IPA, and a non-polar solvent, which is hexane. If you've ever heard the phrase, like dissolves like, the same sentiment holds true here. Water, our most polar solvent, is going to be capable of extracting the most polar components from the polymer. Likewise, our non-polar solvent, hexane, is going to be isolating and extracting all of the non-polar components. So if we go to our water experiment, this is again is going to be a leachable situation. So this is our leachable experiment. We're going to analyze these extracts by LCMS to detect the non-volatile components which it pulls out. This is also going to be the optimal solvent for detecting heavy metals, as well as getting a full elemental screen that we will run by ICP-MS. IPA is our kind of middle solvent, and this is going to extract a wide range of polar to non-polar components. Thus, we're going to use 2 different methods to detect what components were extracted, LCMS as well as GCMS. Finally, hexane, which is our non-polar solvent. This will extract the non-polar materials, and these are they tend to be volatile components, so we'll analyze these using GCMS. So how are these samples actually extracted? For this, we took one whole medical device, so this will include both the layers, and we put it in the appropriate solvents, and we used a fixed ratio of 0.2 mils of solvent per 1 gram of sample. And as you saw in the previous slide, we'll be preparing a method blank alongside the actual sample extractions. This will allow us to account for any signals that we detect that are due to the solvent, materials, or containers that are involved in the extraction process. We'll get our samples ready to go, and then we'll heat them for 72 hours at 50 degrees C, and we'll note any visual changes such as degradation or swelling. We'll then take an aliquot of all of those fractions and analyze them by the appropriate techniques. We'll also take a little bit of that solvent and dry it down to a residue to see what is the total percent dissolved solids, so we can calculate how many milligrams or micrograms of material could be extracted from this medical device. So what can we expect to see? The number one thing we're usually looking for here are polymer additives, which can include plasticizers, which add flexibility to polymers, UV stabilizers and antioxidants to prevent them from degrading under UV conditions, and thermal stabilizers, which protect them against high heat. We can often see polymer fragments or small oligomers, as well as other degradation products of the polymer. And depending on how the sample swells, we may be able to detect any sort of other inorganic fillers. So these may be left at the bottom of the extraction vessel for inorganics like talc, silica, or calcium carbonate. Since these are not soluble species, we're going to have to use some different techniques to analyze them, and we'll use our old friends FTIR and EDS to determine their chemical family and elemental composition respectively. In terms of what we're detecting and what we run alongside our samples, again, we use a mass spectral based technique for all the identifications, otherwise identification can be very difficult. We also analyze groups of reference standards alongside our samples so we can provide semi-quantitative data for materials or components that we detect. These can include, again, UV inhibitors or antioxidants, plasticizers, polychlorinated biphenyls, polyaromatic hydrocarbons, and volatile organic compounds. So here we see some examples of things that we see commonly. Again, the 3 at the top with green stars, you'll notice that they contain a lot of nitrogens and oxygens. These are going to be susceptible to taking on a charge and are more likely to be detected by LCMS because they're more polar and will be extracted with water or IPA. The 4 components you see on the bottom are more nonpolar, so these are going to be more likely to be isolated from our IPA or hexane, and these are going to be detected by GCMS. So taking a look at some of the chromatograms from our IPA extract of this medical device, on the top we have our method blank, followed by our aromatic hydrocarbon standard, our phthalate standard, our PCB standard, and then finally our sample extract. We don't have time to go through all the data, but the takeaway message here is we looked at all these different peaks and we're able to assign most of them to siloxane-related species, which again, after our first two case studies, we can determine that this comes from the soft part of the medical device. We did detect some other unique components aside from the siloxane-related species. One of them was tributylphosphine oxide. This was detected at a trace level and is a common reaction byproduct that can be associated with the catalysis of this material. We also detected a polycarbonate fragment. So if you see that structure down the bottom right, if you have some chemical intuition and you remember our structures from the first case study, it should look pretty familiar. So that's a BPA fragment. Looking at some LCMS data, again, we have a method blank, and then we have our sample that was extracted in IPA. Again, after analysis, we see that all these prominent peaks are all siloxane related, which again is going to be consistent with our FTIR as well as our GCMS findings. But we also see this other component, 1-dibutyl-phosphoryl-butane. We'll discuss this a little bit more in the next slide. 1-dibutyl-phosphoryl-butane is actually the same exact thing as tributylphosphine oxide. So as these molecules get more complex, there become more and more different ways to name them. That's why it's important to register or know their CAS registry numbers. One compound can go by multiple different names, but it will only have one CAS number, so it's usually the best way to reference materials or look things up. We also detected palmitic and stearic acid. So these are fatty acids that are typically found together, and they're common processing aids. We also detected 2 UV stabilizers or antioxidants in the forms of Cyasorb UV2908, as well as Ergonox 1076. These were also confirmed to be extracted from the sample through analysis of analytical grade reference standards. Finally, we'll take a look at some example ICP-MS data. So again, this is going to be from our aqueous-based extraction. So you have to keep in mind that each of these solvents is designed to isolate and extract certain materials from the polymer. Water is going to be the best solvent to pull out things like heavy metals. So here we take a look at our example data, and we see that no heavy metals were detected, so we should get some nice warm fuzzy feelings. So what are the takeaway messages from our extractable and leachable experiments? We're mostly detecting siloxane-related peaks by both GCMS and LCMS. We were able to detect tributylphosphine oxide by both GCMS as well as LCMS. We detected some processing aids or fatty acids, as well as 2 UV inhibitors and antioxidants. ICP-MS did not detect any heavy metals, so now this client can take this data to a toxicologist and determine the impacts of this extractable and leachable experiments. So paired with our first 2 case studies, we can also conclude that these additives are likely attributed to the hard part of the medical device. With that, we thank you for your attention, and we'd be happy to answer any questions.